practical series. Myself Prachi Joshi from Department of Pharmaceutical Technology. So today we'll be learning about one of the micro practicals, that is to perform biochemical test of given sample. So here we'll be performing citrate utilization test, which is also known as Simon's citrate test, because we are utilizing Simon's citrate agar. Now this citrate utilization test is a part of IMVIC test, which is also known as indole methyl Vogue Provosker and citrate test. Now, what is the principle and why we need to do this? So first of all, the principle is the ability of the microorganism to utilize the citrate as a source of energy. Now, they utilize it for growth. So if the microorganism grows, then it also indicates that it has utilized the citrate. Now, the agar medium which we have utilized contains two things, that is citrate and inorganic ammonium salt. Now the citrate is basically the source of energy of carbon dioxide. Now inorganic ammonium salt is the source of nitrogen into it. Now here what happens when the growth is indicated, it indicates that citrate is utilized as we have discussed. So the citrate is basically the intermediate metabolite of Krebs test or Krebs cycle, you can say. Now here what happens, citrate is breaking down with the help of enzyme citrase into oxaloacetate and acetate. Now this oxaloacetate again breaks down into pyruvate and carbon dioxide. Now this carbon dioxide metabolizes the ammonium salts, and this ammonium salts form ammonia, or you can say sodium carbonate. Now because of these two products, what happens is the alkalinity of the medium is increased, and due to this there is a shift in pH. So because of this shift in pH, as you can see, as I have written that bromothymol blue in the medium converts its color, or you can say change its color from green to blue. This indicates what? That there is positive citrate test. This positive citrate test indicates what? That utilization of citrate by the microorganism for the growth. So it is also used for differentiation of different organisms like Enterobacteriaceae. So for two things it is utilized. First, as the ability or to determine the source of energy, you can say, or how much amount of energy is being utilized for the growth of microorganism, and second is to differentiate from different Enterobacteriaceae. So thank you. The procedural part will be explained by Ms. Shital Trivedi.
Hello everyone, I will show you how to perform one of the biochemical tests, that is citrate utilization test, which is also one of the tests of the IMVIC group of biochemical tests that is used to differentiate or identify the microorganism of the Enterobacteriaceae family. Okay, so to perform this test, let's see what are the requirements. So we require a septic area, or we can create the environment between two flames. So this kind of arrangement is required. We require test sample or the organisms which are need to be identified based on this test. We require specific medium, that is Simon's citrate agar medium, and for that we can also perform positive test and negative test, means the control, positive control and negative control, and one test tube that is to test the test organism. So we required this kind of media in form of slant, right. So Simon's citrate agar media in form of slant. We required IPA cotton balls to wipe the surface between these two flame, waste beaker to collect the waste items. We require nichrome wire loop to streak or stab the test organism into the slant of the culture medium that we are going to use. So let's see how to perform this practical. Okay, so first step, that is to clean the surface area between these two flame. So that is with the help of cotton balls dipped in IPA, which act as a disinfectant. Then flame sterilize the area like this. Okay, so now area is ready. Sterilize the loop. Okay, let it cool. Now here you can see this Simon citrate agar medium. As per the formula, it contains sodium citrate as only carbon source. So this taste identify the organisms which are capable to utilize this sodium citrate as their sole carbon source. This medium contains bromothymol blue, and the pH of the medium is 6.9, and at this pH look at the color of the medium, it is green in color. Okay, so now the loop is cool. We will take the culture aseptically. Close the culture tube, put it back. Now I will show you how to streak this taste organism into the slant. So for that, sterilize the neck. Now slowly you hold the slant tube like this and just okay, now insert streak the surface of the slant like this, then close the slant tube. So this is called inoculation. So inoculation of the taste organism into the Simon citrate agar medium, that is over the slant of this medium. So likewise we can have positive control and negative control. The positive control slant is to be inoculated with the organism like Klebsiella pneumoniae, which gives this taste positive, means this type of organisms are capable to utilize sodium citrate as a carbon source, and negative control that can be inoculated by the organisms like E. coli. These organisms are not capable to utilize sodium citrate. So this organism will give this taste negative. We can perform simultaneously inoculation of the positive control and negative control. So now after inoculation of all three taste tubes that is containing slant of the Simon citrate agar medium, these taste tubes are incubated at 35 to 37 degrees centigrade for 24 hours in incubator, and after that incubation period now the slants will be observed. Okay, so after incubation period, positive control slant will turn into blue color, negative control slant will remain as such, and if the slant of the Simon's agar medium that is inoculated with taste organism, and after incubation if we observe this kind of observation, means there is no change in the color of the slant and no growth over the strict surface of the slant, then we can say that the taste organisms are giving this taste negative. That means the taste organisms are not capable of utilizing sodium citrate as a sole carbon source, because these organisms lack the specific enzymes, that is citrate permease and citrase. So this is how we can interpret the result of this citrate utilization taste, right. So E. coli, that is the organism that gives this taste negative. So biochemical tests are used to identify the metabolic reaction that occurs in the biological organisms, and these organisms involve specific type of types of enzymes which are responsible for the utilization of the particular source of the nutrient and generate the specific type of metabolic product. So, we can identify specific type of metabolic pathway by biochemical taste. So, biochemical taste are very important for the identification of the microorganism. So, that is all about this taste, that is citrate utilization taste. Thank you so much.